Good morning, everyone, and thank you for being here today. I'm delighted to welcome you to this presentation. Today, we'll be discussing visual inspection of injections for visible particles. This session will explore the methods and importance of inspecting injections visually to ensure safety and quality. We'll cover the key techniques, standards, and best practices involved in this crucial aspect of pharmaceutical quality control. Let's get started and delve into this vital topic. Let's talk about the importance of injectable products being free from visible particulate matter. Particulates are basically extraneous, undissolved particles that can be present in the solution, and it's important to note that gas bubbles are excluded from this definition. These particles can come from various sources, like glass shards or rubber fragments, and they pose significant risks if they enter the bloodstream. For example, glass particles in a vial can lead to embolism, which is a serious health concern. That's why visual inspection is such a critical step in quality control before the product is released to the market. It helps us identify and remove any visible particles that shouldn't be there. Ensuring the product is particle-free is not just good practice. It's actually required by regulatory agencies like the USB, FDA, and EU GMP. These standards help us maintain the safety and efficacy of the injectable products we produce. So, thorough visual inspection isn't just a formality, it's a vital safeguard for patient safety. When we talk about something being essentially free, we mean that during a validated inspection, no visible particles are detected. This isn't just a casual observation. It's based on rigorous testing methods like 100% inspection and acceptance sampling using an AQL or acceptable quality level. For example, if you have a batch of 10,000 vials, only a very small number of defects are allowed, according to the AQL standards. Now, it's important to understand that particles can come from different sources. There are extrinsic particles, which are foreign contaminants like dust or fibers that come from outside the product. Then there are intrinsic particles, which originate from the manufacturing process or the materials used, such as glass flakes or rubber fragments. Lastly, some particles are inherent, meaning they are related to the formulation itself, like protein aggregates. Connecting this to the overall theme, maintaining an essentially free status is crucial for ensuring product safety and quality, especially in sensitive applications like pharmaceuticals or medical devices. It's about controlling and understanding the different types of particles to guarantee the integrity of the final product. Let's take a closer look at patient risk in relation to particles. Particles in medications or solutions can potentially cause serious issues. For example, they might lead to embolism, which is when a blockage occurs in blood vessels. They can also trigger inflammation, which can damage tissues and organs. In some cases, particles can cause direct organ damage, depending on their size and where they travel in the body. It's important to note that certain patients are at higher risk. Neonates, for instance, have more delicate systems, making them more vulnerable to these particles. Similarly, patients receiving IV or intrathecal administration are at increased risk because the particles bypass some of the body's natural filtering mechanisms. For example, a fiber particle could increase the risk of a pulmonary embolism if it travels to the lungs. So, understanding these risks helps us better assess and improve medication safety for our patients. Now, let's talk about the nature of visual inspection. It's important to understand that this process is probabilistic, meaning we can't guarantee 100% detection every single time. Detection depends on several factors, including the particle size, color, and shape. For example, larger or more contrasting particles are easier to spot. The type of container also plays a role, as some containers make inspection more challenging than others. Additionally, the clarity of the product itself can influence how easily defects or particles can be seen. For instance, a transparent solution makes detection much simpler because you can see through it clearly. On the other hand, an opaque suspension can make inspection more difficult because visibility is limited. When it comes to detection capability, we generally have a high probability of catching particles that are 200 micrometers or larger. Fibers, which are typically around 500 micrometers, are a bit harder to detect reliably. This has an important implication. Smaller particles may often go unnoticed during visual inspection, so we need to be aware that it's not a foolproof method. Overall, understanding these nuances helps us better evaluate the limitations and effectiveness of visual inspection in our quality control processes. Let's walk through the inspection process flow. First, we have 100% inspection, which means every item is checked thoroughly to catch any issues early. Next is acceptance sampling, where we review a sample of units to determine if the batch meets quality standards. After that, we move to investigation and trending, which involves analyzing data over time to identify patterns or recurring problems. This step helps us understand root causes and prevent future issues. Finally, we focus on continuous improvement, using the insights gained to enhance our processes, increase quality, and reduce waste. Together, these steps create a robust system for maintaining high standards and driving ongoing excellence. Now, let's look at the different types of inspection we use. First, there's the manual visual inspection. This is performed by trained operators who carefully examine each sample. To ensure consistency, we conduct these inspections under controlled conditions, specifically with lighting around 2000 to 3750 lux and using a black and white background to improve contrast. The typical procedure involves swirling the vial, observing it against the background, rotating it to check all angles, and then inspecting for any anomalies. Moving on, we also have semi-automated and fully automated inspection methods. The semi-automated process combines conveyor systems with human inspection, allowing for higher throughput while still relying on human judgment. On the other hand, fully automated inspection, such as automated visual inspection or AVI, uses advanced cameras and sensors. These systems can automatically reject samples based on programmed criteria, which helps improve consistency and efficiency. For example, the machine might detect moving particles through light scattering techniques, flagging any irregularities that need further review. Overall, these different inspection methods are designed to ensure product quality while balancing accuracy, speed, and resource use. Now, let's talk about qualification and validation. It's essential to demonstrate our detection capability, which means showing that our system can reliably spot defects. Consistency is also key, making sure the process performs the same way every time. To do this, we use defect standards, like seeded particles, as a benchmark. For example, we might seed a vial with a 150 micron glass particle to test whether the inspector can detect it accurately. Speaking of inspectors, their qualification is equally important. They need proper training, which starts with a visual acuity test to ensure their eyesight is sharp enough. Requalification is also necessary periodically to maintain high standards. And we can't forget about fatigue. It's crucial to manage fatigue properly because tired eyes can lead to missed defects. For instance, operators are often rotated every 30 to 60 minutes to keep alertness high. All of these steps help us maintain a robust inspection process, ensuring product quality and safety. When it comes to difficult-to-inspect products, or DIP, we're talking about some of the more challenging types to evaluate visually. Examples include suspensions, emulsions, and lyophilized powders. These products tend to be tricky because their physical characteristics can obscure defects or impurities. One major challenge we face is turbidity, which makes the product cloudy and hard to see through. Opacity is another issue, especially with products that are naturally opaque or formulated to be so, making quality checks more complex. Plus, when these products are stored in complex containers, it can be even more difficult to perform thorough inspections without specialized techniques. So, what's the solution? Enhancing lighting is a key step. Using better illumination can reveal issues that might otherwise go unnoticed. Additionally, employing alternative inspection methods, like using different angles, maybe even some non-visible light techniques, can help us get a clearer view and ensure product quality. Overall, recognizing these challenges and applying the right solutions is essential for maintaining high standards, especially when dealing with these complex, difficult-to-inspect products. Now, let's move on to post-inspection verification. It's important to confirm that any units we've rejected are properly segregated to prevent mix-ups later on. This step ensures that rejected items don't accidentally get back into the production flow. We also need to recheck borderline units, which means doing re-inspections where applicable, especially for those that are close to the acceptance criteria. For example, if a vial is flagged for fiber, it should be sent for a secondary inspection before making the final rejection decision. This extra step helps us avoid unnecessary rejections and ensures accuracy. Next, we have AQL, or Acceptance Quality Limit Sampling. This involves taking a statistical sample from the inspected batch, say, sampling 200 vials, and analyzing it for defects. The goal here is to ensure that our inspection process is effective overall. If the defects found in the sample stay within the AQL limits, we can be confident that the entire batch meets quality standards. Finally, let's discuss defect classification. We categorize defects into critical, major, and minor. Critical defects, like visible particles, can significantly impact the batch's safety or efficacy. Major defects are cosmetic issues that might affect the inspection process, but aren't necessarily safety hazards. Minor defects are non-critical appearance issues that don't affect the product's function or safety. Understanding these classifications helps us prioritize our response and maintain high-quality control standards across our processes. Now, let's look at how trending and data analysis play a vital role in our process. By monitoring defect types and their frequency, we can quickly spot patterns and identify recurring contamination sources. For example, if we notice an increase in glass particles, that would prompt us to investigate our vial supplier more thoroughly. Moving on to investigation and CAPA. This is where root cause analysis comes into play. We need to understand exactly what's causing these defects so we can implement effective corrective and preventive actions. This step is crucial to prevent issues from recurring and to keep our products safe. Next, documentation and batch release are key components. We record all inspection results, rejects, and rechecks meticulously. Before releasing each batch, QA conducts a thorough review to ensure everything meets quality standards. Ultimately, quality control ensures that each inspected vial or ampule is essentially free of visible particles. This is fundamental not only for patient safety, but also to maintain regulatory compliance. Consistent quality control measures help us deliver safe, reliable products every time. Let's talk about lifecycle and continuous improvement. As USB 1790 emphasizes, our focus isn't just on detecting defects after they occur, but on preventing them from happening in the first place. Prevention is key to maintaining quality and efficiency. We also need to pay close attention to trending defects over time. By analyzing these trends, we can identify patterns that might point to underlying issues. This proactive approach allows us to address problems before they escalate. Root cause analysis is another critical aspect. When we notice an increase in a specific defect, like fiber defects, we don't just fix the symptom, we dig deeper to find the root cause. For example, an uptick in fiber defects might lead us to review the gowning procedure to see if that's contributing to the problem. The idea is to create a cycle of continuous improvement. By constantly reviewing our processes, trending defects, and addressing root causes, we foster a culture of ongoing enhancement. This approach helps us stay ahead of issues and improve quality throughout the life cycle of our products. For further support, please don't hesitate to reach out. We're here to help and provide any additional information you might need. If you have questions or need clarification, just contact us through the details provided. Thank you so much for your time and attention today. We truly appreciate your interest and engagement. Remember, we're always happy to assist you as you move forward. Thanks again, and have a great day.